We're going to look at how the human body takes this complex architecture, proteins, fats, carbohydrates, and turns it all into microscopic fuel. And we're going to do this mechanism by mechanism.
And, to keep us on track, because this is a really dense physiological system, we should probably introduce the big three.
Right. The macronutrients. Carbohydrates, fats, and proteins.
Almost everything you eat, in a meaningful caloric sense, falls into one of those three categories.
And the body has a very specific, very distinct chemical assembly line for each one.
Oh, absolutely. The machinery for handling a pat of butter is radically different from the machinery for handling a potato.
But, and this is the part that I found fascinating in the source material, despite how different a piece of fat is from a piece of sugar, the chemical reaction that breaks them down is actually identical.
It is. This is a huge point. I think a lot of people miss this.
I want to pause on this because I definitely missed it. I always just assumed that digesting fat was, a totally different chemical universe than digesting sugar.
Well, the tools are different.
What do you mean, the tools?
The enzymes. You use a different tool to cut a diamond than you do to cut wood, right? But the fundamental action, the chemistry that snaps the molecular bond, it's the same.
And that reaction is called hydrolysis.
Hydrolysis.
Let's really dig into this, because this seems like the skeleton key for this entire deep dive. If you understand this one reaction, you basically understand digestion.
You do, but to understand the hammer, you have to understand the nail. You have to understand how plants and animals build these structures in the first place.
Okay, so how are they built?
When a plant builds a starch chain, or your body builds a muscle fiber, it takes these small building blocks, monosaccharides or amino acids, and it links them together.
Right.
But to get them to snap together, the chemistry requires the removal of water.
Remove water? You mean like literally an H2O molecule is ejected from the bond?
Literally. The process is called condensation. Chemically, you take a hydrogen ion, an H plus from one building block, and a hydroxyl ion, an OH minus from the one next to it.
So H and OH.
They combine to form H2O water. That water molecule is removed, and in that empty space, the two blocks bond together. That's how a sugar chain is built. That's how a protein chain is built.
So structurally, a carbohydrate chain or protein fiber is just a whole bunch of simple blocks that are essentially glued together by the act of dehydration.
Precisely. It's dehydration synthesis. So if that's how you build it, digestion is simply the reverse.
You put the water back?
You put the water back. You could call it the revenge of the water if you want.
Yeah.
The digestive enzymes, that's their whole job. They grab an H2O molecule from the digestive juices, they split it back into H plus and OH minus, and they wedge them back in between the building blocks.
And that forces the bond apart.
It breaks the bond by re-adding water. Hydro, meaning water, and lysis, meaning to split. Hydrolysis.
So that's the rule of the game. That's the core principle. Whether it's a massive protein chain in that steak or a glob of fat in some butter, we're just using specific enzymes to shove water back into the structure to break it apart.
That is the universal mechanism. But as you pointed out, the tools, the enzymes, they are highly, highly specific.
Right. You can't use a wrench to unscrew a light bulb.
Exactly. You need the specific enzyme for the specific food. The chemistry is always the same, but the lock is always different.
Okay, so if the chemistry is universal, why is the digestive system so compartmentalized? Why do we need a stomach and a pancreas and a small intestine if it's all just adding water?
Because of the environment, those specific tools need to function. It's all about the conditions. Some tools, some enzymes, need a brutally acidic environment to work. Others would be destroyed by acid and need a neutral or alkaline environment. Some need other chemicals, like bile, to even get started.
So it's an assembly line where every station has a different pH and a different set of specialized workers.
That's a perfect analogy for it.
All right, let's start walking down that line. Let's start with station one, the fuel that powers most of the world. Carbohydrates.
Carbohydrates. This is our bread, our pasta, our fruit, our sugar.
And in the typical human diet, we're really looking at three major sources, right?
That's right. For the most part, it boils down to three. You've got sucrose, that's your common table sugar, cane sugar. You've got lactose, which is the sugar found in milk. And then the really big one, starches.
Grains, potatoes, rice, all the staples.
Right. These are what we call large polysaccharides, just massive, long, branching chains of glucose molecules all linked together.
Now, there is another carbohydrate that we eat a lot of, but the source material says we can't actually digest it, cellulose.
Yes, cellulose. That's dietary fiber.
So the stringy stuff in celery, the tough parts of kale.
Exactly. And chemically, it's very similar to starch. It's also a long chain of glucose. But the way the glucose molecules are linked is slightly different. It's a different kind of bond.
And that small difference matters.
It's everything. Humans, unlike cows or termites or other herbivores, we lack the specific enzyme required to hydrolyze that specific bond in cellulose.
We don't have the key to that particular lock.
We don't. So for us, it's not food. It holds 0 caloric value. It just passes right through the system.
It's just scenic traffic then. It helps keep things moving, but it doesn't actually fuel the engine.
A perfect description. But for the starches we can eat, the process starts way earlier than most people realize. It actually starts before you even swallow.
Phase 1, the mouth.
Right. As you're chewing, your salivary glands, specifically the parotid glands, the big ones in your cheeks, are pumping out an enzyme called ptyalin.
Ptyalin, which is a type of alpha amylase.
It is. And it works fast. I mean, before that bite of food even feels ready to swallow, ptyalin is already on the attack.
What's it doing?
It's hydrolyzing the starch. It's snipping those long chains into shorter bits, mostly into a disaccharide called maltose and some other small glucose polymers.
So if I chew on a saltine cracker and just hold it in my mouth for a minute, that slight sweetness I start to taste, that's not my imagination.
That is absolutely not your imagination. That is exactly what that is. That's hydrolysis happening in real time.
Wow.
The tasteless, complex starch is literally becoming sweet-tasting maltose right there on your tongue. But here's the dramatic part. It's a race against time.
Or, more specifically, a race against acid.
A race against acid. Because the stomach is the next stop, and the stomach is, an acid bath.
Right. You swallow the food, it drops into the stomach. And the stomach acid, the hydrochloric acid, is incredibly intense.
Which brings up a contradiction I noticed in the notes. We just established that ptyalin, the saliva enzyme, works great on starch. But acid completely destroys ptyalin. It blocks its activity.
So by swallowing, aren't we essentially just turning off our own digestion almost immediately?
You are eventually, but it's not an instantaneous shutdown. And this is due to a really cool physics dynamic called the food bolus effect.
The food bolus.
Yeah. When you swallow food, it doesn't just splash into the stomach like water. It lands in a clump, a bolus, and the acid attacks the outside of that clump first.
Ah, so the inside is protected for a while.
The inside is still at a neutral pH. It's shielded by the outer layers of food.
So the little saliva enzyme is hiding inside this food ball, still working frantically.
Exactly. It's a race. The ptyalin is in the center, just furiously chopping up starch, while the stomach acid is slowly burning its way in from the outside.
How long does that race last?
For about an hour, give or take. Once the acid finally penetrates to the center of the bolus and the overall pH drops below 4.0, the amylase activity stops cold.
So how much work did it actually get done before the acid shut it all down?
A surprising amount, actually. The textbook says on average about 30 to 40% of the starches are hydrolyzed before the stomach secretions completely halt the process.
Nearly half the job is done just by saliva and that initial stomach holding time. That's incredibly efficient.
It is. But then, of course, the food, which is now a soupy mixture called chyme, moves from the stomach into the small intestine, and this is where the big guns come out.
Phase 2, pancreatic digestion.
The pancreas secretes its own pancreatic amylase, and this stuff is, well, it's extremely potent.
So it's different from the saliva version.
Chemically, it's almost identical to the saliva version, but there's a lot more of it, and it's several times more powerful. It's the industrial-grade version.
The heavy hitter.
Absolutely. Within just 15 to 30 minutes of that chyme emptying into the duodenum, that's the very first part of the small intestine, the pancreatic amylase has done its job.
And what's the result?
Virtually all the remaining carbohydrates are now digested. They're broken down into maltose or other small glucose polymers.
Okay, so we've taken the massive complex starch and we've broken it down into these small clusters. But we're not at the single molecule level yet, are we? We can't absorb maltose directly.
No, we can't. Maltose is still 2 glucose units stuck together. It's a disaccharide. Our bodies can only absorb monosaccharides, single sugars.
So there has to be one more step.
There is. And that leads us to phase 3, the brush border.
Brush border. I love that term. It sounds like a geographic frontier, like a place on a map.
It essentially is a frontier. We're talking about the inner lining of the small intestine. This lining isn't smooth. It's covered in these tiny finger-like projections called villi.
Okay.
And the cells that make up those villi, called enterocytes, their surface looks like a brush because of thousands of even tinier protrusions called microvilli.
So it's like a deep pile carpet.
An excellent analogy. And embedded right there in that carpet, right in the brush, are the final enzymes,
So they're not floating around in the juice, they're physically attached to the wall?
They are attached to the cell membrane. They're basically waiting there to catch these sugars as they bump up against the wall.
They're specialists.
The ultimate specialists. There are 4 of them, each with a very specific job.
Let's run down the list. Who are they?
Okay, first you have lactase. Its one and only job is to split lactose, the milk sugar.
And it splits it into...
Into 1 molecule of galactose and 1 molecule of glucose.
Okay, and if you don't have enough of that particular enzyme...
Then you're lactose intolerant. The lactose can't be split, so it stays in your gut. The bacteria down the line have a party, they produce gas, and, well, you have a very bad day.
Got it.
Okay.
Next enzyme.
And next up is sucrase. It takes sucrose, your basic table sugar, and it splits that into 1 molecule of fructose and 1 of glucose.
Simple enough. And what about the starches?
Well, remember, the amylase already broke the starches down into maltose, so the brush border has maltase.
Maltase for maltose. Makes sense.
And it splits maltose into 2 glucose molecules. Glucose plus glucose.
So that's it.
Almost. For any of those small leftover glucose polymers that weren't quite maltose, there's a final cleanup enzyme called alpha-dextrinase. It handles the odds and ends.
So it's a clean sweep. Every single carbohydrate, no matter where it came from, gets broken down to a single sugar molecule.
Everything. And if you look at the final tally, the end result of all carbohydrate digestion is monosaccharides, and the winner, by an absolute landslide, is glucose.
How much of the final product is glucose?
The sources say over 80% of the final absorbed product is glucose. Galactose and fructose usually represent less than 10% each.
So really, when we talk about blood sugar, we're almost exclusively talking about the glucose that results from this entire process.
That's right. The body converts almost everything into glucose for its primary fuel.
So that's carbohydrates, done, dusted, turned into immediately usable fuel. Now let's pivot to the second major category, the bodybuilders. Proteins.
Right, and this is, chemically speaking, a much tougher nut to crack.
Why is that?
Proteins are these incredibly long, folded chains of amino acids, and they're held together by something called peptide linkages. These bonds are very strong.
And unlike with carbohydrates, the mouth doesn't really do anything here, correct? Saliva doesn't digest meat.
Correct. There are no proteolytic enzymes in saliva. Protein digestion begins in the stomach, phase 1. And this is where the stomach's famously acidic environment becomes a feature, not a bug.
We usually just think of stomach acid as, burning, heartburn, indigestion.
Yeah.
But it has a critical purpose.
It has a crucial 2-fold purpose. The main enzyme for protein digestion in the stomach is pepsin.
Pepsin.
And pepsin is a very unusual enzyme. It thrives in violence. It is most active at a pH of 2.0 to 3.0.
That is incredibly acidic. That's like battery acid level.
It is. And if the pH in the stomach rises above 5.0, pepsin basically just falls asleep. It becomes completely inactive.
So the stomach glands have to secrete huge amounts of hydrochloric acid just to create the right working conditions for this one enzyme.
Precisely. That's the first purpose of the acid, to activate pepsin.
But why? Why design a system that requires such a dangerous corrosive acid? Why not just use an enzyme that works at a neutral pH, like in the mouth?
Because of what it has to digest, specifically a protein called collagen.
Collagen, that's the connective tissue in meat, right? The gristle, the tough stuff.
Exactly. The stuff that holds the muscle fibers of the meat together. Collagen is an albuminoid type of protein that is almost totally resistant to most other digestive enzymes.
But not to pepsin.
But not to pepsin. Pepsin is uniquely capable of digesting collagen. This is its superpower.
So if you didn't have pepsin.
If you didn't have pepsin, or if your stomach acid was too weak to activate it, the other digestive enzymes further down in the intestine wouldn't be able to effectively penetrate the meat fibers.
They couldn't get in.
The meat would stay clumped together, held fast by the collagen. You'd get very, very poor protein digestion from meat.
So pepsin's the can opener. It cracks open the safe so the other enzymes can get to the money inside.
That is the perfect analogy. But, of course, there's a flip side to having an enzyme that's designed to dissolve meat.
Right. You are made of meat. The stomach wall itself is protein.
This is the constant, dangerous tension of the stomach. It is holding a vat of corrosive acid and an enzyme designed to liquefy protein while trying desperately not to liquefy itself.
How does it manage that?
It protects itself by secreting a very thick alkaline layer of mucus. It's a physical and chemical barrier. But if that mucus barrier fails for any reason,
You get a stomach ulcer. You literally start digesting your own stomach wall.
Exactly. So pepsin does the initial rough chopping. It breaks the collagen, and it starts splitting the big protein chains into smaller fragments, things called proteases and peptones.
So it doesn't finish the job.
Not even close.
Oh.
It doesn't break them down into individual amino acids. It just creates a chaotic soup of smaller protein pieces. And when that chyme is ready, the stomach squirts it into the small intestine.
Phase 2, the pancreatic enzymes. The pancreas really is the hero of digestion, isn't it? It seemed to handle everything.
It's the absolute workhorse. As soon as that acidic chyme enters the duodenum, the pancreas releases a whole cocktail of powerful proteolytic enzymes.
A whole team.
A whole team. You have trypsin and chymotrypsin. They act like very powerful scissors, splitting the large protein fragments into much smaller polypeptides.
But still not individual amino acids yet.
For the most part, not yet. Then you have another one called carboxypolypeptidase. This one is more precise. It goes to one specific end of the polypeptide chain, the carboxyl end, and it cleaves off individual amino acids, one by one.
So it's snipping them off the end of the chain like beads off a string.
Exactly. And there's one more specialist in the pancreatic juice, elastase. As the name implies, it specifically digests elastin fibers, which is the other type of connective tissue holding meats together.
So after this pancreatic blitz is over, what are we left with?
We're left with mostly very small chains, mostly dipeptides and tripeptides. That's 2 or 3 amino acids linked together. Only a small percentage are fully free single amino acids at this stage.
So we are very close, but we're not quite at the finish line. We need to get down to single amino acids to absorb them efficiently.
And that brings us back once again to the enterocytes, the intestinal cells. This is phase 3. And this is fascinating because the final step of digestion actually happens on and inside the cell.
Wait, inside the cell, not in the tube of the intestine.
Both. First, the microvilli, that brush border, has its own peptidases protruding from it, things like a monopoly peptidase and various dipeptidases.
And they keep splitting the peptides as they touch the cell wall.
Correct. But even then, some dipeptides and tripeptides actually get transported into the cell while they're still linked together.
They sneak in as a duo or a trio.
They get in, but the cell is waiting for them. Once they are inside the cytosol, that's the fluid inside the cell, there are other specific cytosolic peptidases that rush in and break those last remaining bonds.
It's the final cleanup crew, inside the house.
The final cleanup crew. Within minutes, virtually all the last dipeptides and tripeptides are split. So the final output, the product that leaves the other side of the cell and enters the blood,
is what?
More than 99% single amino acids.
It's just ruthless efficiency.
Mm-hmm.
The body absolutely refuses to let a chain survive. It only wants the individual links.
It has to have the individual links.
Yeah.
It can't use cow protein to build human muscle. It needs to break the cow protein down to the universal bricks, the amino acids, and then use its own DNA blueprint to rebuild them into human structures.
That makes perfect sense. Okay, so we've done bread, we've done steak, now we have to do the butter. Let's talk about the trickiest one, fats.
Section 3, the digestion of fats. And you're right, this is the most challenging one, but not chemically. It's a physics problem.
It's the oil and water problem. Our entire digestive system, all the juices, they're water-based.
Exactly. And the main type of fat we eat is triglycerides. You might also see them called neutral fats. That's 3 fatty acid molecules attached to a single glycerol backbone.
And the problem is, in the watery environment of the stomach and intestine, these fats just clump together into big globs.
Right, and our enzymes, which are water-soluble, can only attack the surface of that fat glob.
So if you have a big ball of fat, the enzyme can't get to the molecules in the middle. It would take way too long.
Way too long. If you didn't have a way to break that glob up, you simply couldn't digest it fast enough. You'd just excrete most of the fat you ate.
So the first step has to be breaking up the globs. Phase 1, emulsification.
Emulsification. And this is where a whole new organ gets involved, the liver.
By way of the gallbladder.
By way of the gallbladder, which stores and concentrates the liver's product. The liver secretes bile. Now, it's really important to note, bile does not contain any enzymes. It doesn't chemically digest anything itself.
So what's in it?
It contains 2 key ingredients, bile salts and a phospholipid called lecithin.
And what do?
They act as detergents. Literally the same principle as dish soap cleaning a greasy pan. They decrease the surface tension of the fat.
How does that work?
It allows the fat to break apart into tiny droplets when it's agitated.
And the agitation is just the normal churning movement of the stomach and intestine.
Exactly. The bile salts coat the fat, The gut contracts, and these large globules are broken into microscopic sizes.
How much of a difference does this mechanical breakdown actually make?
According to the source, it increases the total surface area of the fat by as much as 1,000-fold.
Wow, 1,000 times more surface area for the enzymes to attack.
Which is absolutely critical because now the main fat-digesting enzyme, pancreatic lipase, can get to work.
This is phase 2 then, the enzymatic part.
Right, and the pancreas secretes pancreatic lipase in enormous quantities. It's one of the most abundant enzymes. It just swarms all that newly exposed surface area and starts hydrolyzing the triglycerides.
And what does it split them into?
It splits them into two main products, free fatty acids and two monoglycerides.
Okay, but here's the problem I see now. We've split them, but they're still fats. They still don't want to dissolve in the watery chyme to get over to the intestinal wall to be absorbed. They're just going to float and try to clump back together.
That is the crucial transport problem, and the body's solution to this is one of the most elegant mechanisms in all of physiology. Phase 3, the micelles.
The ferry system. I remember this from the notes. This is incredible.
It is. Those bile salts, remember them from the emulsification step? They have a superpower. They are what we call amphipathic.
Amphipathic, what does that mean?
It means one side of the molecule is fat-soluble, lipophilic, and the other side is water-soluble, hydrophilic. It loves both fat and water. So they spontaneously arrange themselves in the water, clumping together to form these tiny spheres called micelles.
A sphere.
A tiny sphere, only about 3 to 6 nanometers in diameter, and they arrange themselves with their fat-loving sterile nucleus in the center and their water-loving polar groups on the outside facing the water.
So it's like a tiny little submarine. It's got a fatty core on the inside and a water-friendly shell on the outside.
That's a perfect way to put it. And they trap the free fatty acids and the monoglycerides inside that fatty core. They hide them from the water.
And then they ferry them.
They ferry them. They shuttle the fats through the watery chyme right up to the brush border of the epithelial cells.
Then when they get there, what happens?
They drop off their passengers. The fatty acids and monoglycerides are lipid-soluble, and so is the cell membrane, so they just diffuse right out of the micelle and into the cell. They slide right in.
And the micelle itself, the bile salts.
They stay behind in the chyme. The ferry boat turns around to go back and pick up more passengers.
It's a reusable shuttle service.
It is. The bile salts are used over and over again.
How important is this ferry system? I mean, what if we didn't have bile salts to form micelles?
Without micelles, you would only absorb about 40% of the fats you eat. The rest would be lost in the feces.
And with micelles?
With them, you absorb about 97% of dietary fat.
That is the difference between nourishment and starvation, basically.
Or at the very least, the difference between health and severe metabolic problems and vitamin deficiencies.
Okay, so we've broken everything down, we've chopped all the wood. Now we have to actually bring it into the house. We need to talk about the process of absorption.
The anatomical basis of absorption.
And this is where we get into that tennis court analogy, because the intestine isn't just a smooth, simple pipe.
Not at all. If the small intestine were just a smooth tube, it wouldn't have nearly enough surface area to absorb all these nutrients before the food passed through. So evolutionists solved this problem with geometry.
By folding.
By folding on multiple scales. First, you have the folds of Kerckering. These are large circular folds that you can see with the naked eye. They run around the inside of the intestine. They alone triple the surface area.
Okay, so that's a 3x increase.
Then, covering those folds, you have the villi. Millions and millions of tiny finger-like projections, about a millimeter long. They give the surface a velvety texture that increases the area another tenfold.
And then on the villi themselves, we mentioned this before.
On the surface of every cell on every villus, you have the microvilli, the brush border we talked about. And that increases the surface area another 20-fold.
So let's do the math. 3 times 10 times 20.
It comes out to a 1,000-fold total increase in absorptive area compared to what a simple, smooth tube would have.
And what does that translate to in real-world terms?
It creates a total surface area of about 250 square meters, roughly the size of a singles tennis court.
A tennis court packed inside your abdomen? That is unbelievable.
It is an incredible feat of biological engineering. And inside each of those tiny villi, there's a whole plumbing system ready to go.
Right. There's a vascular system, the blood capillaries, to take up the carbs and proteins.
And in the very center of the villus, there's a vessel called the central lacteal. That's a lymphatic vessel, specifically for taking up the fats.
And the source material mentioned something really cool about actin filaments inside the microvilla. They're not just static, are they?
Oh, this is amazing. No, they are not static hairs. They have actin filaments extending down from them into the cell body that allow them to contract and extend. They move rhythmically.
They wave, like seaweed in the current.
More like they pulse. And this constant movement keeps them exposed to new intestinal fluid. It prevents the fluid right next to the wall from becoming stagnant and depleted of nutrients.
It's actively stirring the water at a microscopic level to speed up absorption.
That's exactly what it's doing.
It's alive. That is wild. Okay, so we have the surface area. Now let's talk about the actual mechanics of moving things across that massive border. And it seems like water and sodium are the leaders of this dance.
They absolutely are. Let's start with water. It's called osmotic absorption. And the key thing to know is that water moves entirely by diffusion. It just follows the laws of osmosis.
So, simply put, where the salt goes, water follows.
Always. The body doesn't have a water pump. To move water, you have to first move solute ions. If you pump ions out of the gut and into the cells, water is magnetically pulled right along with them to keep the concentration balanced.
So to absorb water, you have to absorb ions. And the king of all ions here is sodium.
The king. I want to focus on this because reading the text, it seems like sodium is the engine of almost all absorption. It drives everything else.
It truly is. The mechanism is a classic example of active transport, so this costs energy.
This costs a lot of energy. On the basalatal membrane of the intestinal cell, that's the side facing away from the gut towards the blood, there are incredibly powerful pumps, the sodium-potassium ATPase pumps.
And what are they doing?
They are constantly actively pumping sodium OUT of the cell and into the space around it, where it then enters the blood.
Which means the inside of the cell becomes very low in sodium.
Correct. The concentration drops to about 50 milliequivalents per liter, compared to about 142 in the gut lumen. This creates a steep electrochemical gradient.
So the sodium in the gut desperately wants to rush into the low-sodium cell. It's like a crowd of people pressing against a closed door.
It is. There's immense pressure for sodium to move from the gut into the cell. And the body brilliantly harnesses that pressure. That inward rush of sodium provides the energy to drag other things along with it.
It's the motive force for so much else. And this is controlled by hormones too, right? Aldosterone.
Yes, absolutely. If you become dehydrated, your adrenal glands secrete the hormone aldosterone. And one of its main jobs is to go to the intestines and turn up the activity of all these sodium pumps.
So it tells the intestine, do not let a single atom of sodium or chloride escape.
Conserve salt at all costs. Because by saving the salt, you save the water that follows it. This is especially crucial for the colon's ability to conserve water.
So sodium is driving the bus. Yeah. What about chloride? It mostly just follows along passively, doesn't it?
For the most part, yes. Sodium ions are positively charged. As they get pumped into the cell, it creates a net positive charge. Chloride ions are negatively charged, so they are pulled along by that electrical gradient to maintain neutrality.
Okay, what about bicarbonate? The pancreas and duodenum secrete a lot of it to neutralize all that stomach acid. Do we get that back, or is it just lost?
We do get it back, but in a very clever, indirect way. We don't absorb the bicarbonate molecule directly. What happens is, when sodium is absorbed, a hydrogen ion, H+, is often secreted into the gut in exchange.
Okay.
That H+ immediately meets the bicarbonate, the HCO3-.
Yeah, there we are.
They form carbonic acid, H2CO3, which is very unstable and almost instantly dissociates into water and carbon dioxide gas, CO2.
Ah, so we reabsorb the bicarbonate by turning it into gas and water.
Precisely. The water is absorbed and the CO2 diffuses into the blood and is eventually just breathed out by the lungs. It's an elegant solution.
Chemistry is magic sometimes. Okay, let's get to the good stuff, the nutrients. How do they hitch a ride on this sodium train?
Section 6. Mechanics of nutrient absorption. Let's go back to glucose. We established it's the main fuel, but glucose is a relatively large molecule, and it's not lipid-soluble. It can't just diffuse into the cell.
It needs a special ticket to get in.
It does. It uses a mechanism called co-transport, or secondary active transport. Remember that massive crowd of sodium ions rushing to get into the cell?
Crowd pressing at the door.
Right. Well, embedded in the cell membrane is a specific carrier protein called SGLT1. And you can think of it like a revolving door with two seats. One seat is for sodium and the other is for glucose.
And it won't turn unless both seats are filled.
It will not work unless both sodium and glucose bind to it simultaneously. The intense desire of the sodium to get into the cell literally provides the force to pull the glucose along with it.
So the glucose gets dragged in, even against its own concentration gradient.
Exactly. It's secondary active transport because the energy wasn't spent directly on moving glucose. The energy was spent creating the sodium vacuum, and the glucose just rides that vacuum wave for free.
And galactose uses the same SGLT1 transporter.
Identical mechanism, but fructose. Fructose is the odd one out.
Fructose doesn't use the sodium bus.
No, it doesn't. It uses a completely different mechanism called facilitated diffusion through a different transporter called GLUT5.
Facilitated diffusion.
Yeah.
So it's not actively pumped.
Correct. It just flows down its concentration gradient from high concentration in the gut to low in the cell. It's a passive process, which means it's much slower.
How much slower?
Its maximum rate of absorption is only about half that of glucose. And once the fructose gets inside the cell, it's usually phosphorylated and then quickly converted into glucose anyway before it enters the blood.
Interesting. And what about proteins, the single amino acids?
A very similar story to glucose. Most amino acids use sodium cotransport. There are at least 5 different types of amino acid transport proteins, but most of them rely on that sodium gradient to pull the amino acid into the cell.
So really, if your sodium pump stopped working, you'd effectively starve, wouldn't you? You wouldn't be able to properly absorb your sugars or your proteins.
You would die very, very quickly. Sodium absorption is the absolute foundation of all nutrient absorption.
Now let's revisit the fats. The Micelle drop them off at the door. They diffuse into the cell. Then what? Do they go straight to the blood like sugar and amino acids?
No. And this is a major, major difference. Once the free fatty acids and monoglycerides are inside the intestinal cell, they're taken up by the smooth endoplasmic reticulum, and the cell rebuilds them.
Rebuilds them. We just spent a huge amount of time explaining how we meticulously broke them down.
I know. Biology is funny that way. But yes, the cell takes those component parts and immediately resynthesizes them back into new triglycerides.
But why? What's the point of that?
It's for packaging and transport. These new triglycerides are then coated with protein, cholesterol, and phospholipids to form little globules called chylomicrons.
Chylomicrons.
And these chylomicrons are way too big to enter the tiny capillaries of the blood system in the villus. So instead, they are ejected out of the other side of the cell and into the central, the lymph vessel.
So fat enters the lymphatic system first. It completely bypasses the liver initially.
Correct. It travels up through the lymphatic system, through the thoracic duct, and is slowly emptied into the large veins near the neck. It enters the general blood circulation much more slowly and diffusely.
Is there an exception to that?
Yeah.
The source mentioned something about butter.
Ah, yes. Short and medium-chain fatty acids, like the ones found in butterfat. They're small enough to be somewhat water-soluble. They don't need the whole Micelle system or to be rebuilt.
So what happens to them?
They can be absorbed directly into the portal blood, just like glucose and amino acids. They go straight to the liver.
So butter is a bit of a speed demon, relatively speaking.
In the world of fats, yes.
Okay, we've digested and absorbed almost everything. But there's still some stuff left over. The indigestible fiber, some water, some dead cells. It all moves into the final stretch of the system, the large intestine.
The colon. And its main job is really twofold: absorption of the remaining water and electrolytes, and storage of feces until defecation.
It's the drying and storage facility.
Essentially. The small intestine absorbs most of the water, but the colon does the final, critical job of drying out the fecal matter. It has a huge capacity. It can absorb 5 to 8 liters of fluid and electrolytes per day.
And if it gets overwhelmed, if too much fluid enters too quickly,
then you get diarrhea. If the volume entering the colon exceeds its absorptive capacity, or if irritation or infection prevents it from absorbing water, that water stays in the feces.
But the colon isn't just a passive dryer. It's a teeming ecosystem.
Bacterial action. This is crucial and often overlooked. The colon is home to trillions of bacteria, the colon bacilli.
And we basically feed them our leftovers.
We feed them the cellulose and other fibers that we couldn't digest. They are able to digest a small amount of it for their own energy, but mostly they just thrive in the warm, nutrient-rich environment we provide. And in return, they give us gifts, vitamins. These bacteria synthesize several crucial vitamins: vitamin K, vitamin B12, thiamine, and riboflavin.
Vitamin K is for blood clotting, right?
Yeah.
It is essential for it. In fact, the amount of vitamin K we get from our diet is often insufficient to maintain normal blood coagulation. We actively rely on these bacteria in our gut to produce enough vitamin K to keep our blood clotting mechanism working properly.
So we are truly symbiotic with our own gut bacteria. We need them to survive.
We absolutely are. Of course, they also produce the less pleasant byproducts: gas, Carbon dioxide, hydrogen gas, and methane are all products of bacterial fermentation.
The less polite side of digestion.
Indeed.
Let's talk about the final product then. Feces. What is it, actually, by composition?
Well, first off, it's mostly water. About 3/4 or 75% water. The solid matter only makes up about 25%. And of that solid quarter, what is it made of?
Okay, here's the breakdown from the text, and it surprises most people. About 30% of the solid matter is dead bacteria.
Wait, a third of solid poop is just dead bacteria from our colon?
Yes. The population turnover down there is absolutely massive. Another 30% is undigested roughage, like that cellulose. About 10 to 20% is fat, and only 2 to 3% is protein. The rest is inorganic matter, like calcium phosphate and shed intestinal cells.
And the color. Why is it typically brown?
That color comes from bilirubin derivatives called stercobilin and urobilin. Bilirubin is a breakdown product of hemoglobin from old red blood cells, and it's excreted by the liver in bile. The bacteria in the gut modify it into these brown pigments.
And the distinctive odor.
That is also entirely the work of the bacteria. Their action produces various byproducts like indole, skatole, mercaptans, and hydrogen sulfide. Those are the compounds responsible for the odor.
Well, on that rather fragrant note.
It's simply chemistry at work.
It is chemistry. So what does this all mean? When we zoom all the way back out from the details, what's the big takeaway?
For me, it's about the staggering, unbelievable efficiency of the system. Think about it. You put this chaotic, random mix of biological material, a burger, a salad, a soda, into one end.
Right.
And through this perfectly timed series of chemical baths, massive surface area multiplication, and incredibly sophisticated active pumping mechanisms, the body captures 99% of the useful value.
It extracts the signal from the noise with near perfection.
It does. It returns the water to the molecules through hydrolysis. It breaks them down to their absolute elemental units, and then it absorbs those units and rebuilds you out of them. We are literally reconstructed daily from these broken down chains of other organisms.
It really redefines that old saying, you are what you eat.
Yeah.
You are what you successfully hydrolyze and absorb.
Precise.
Here's a final thought for you to chew on then. We talked about those bacteria in the colon. We provide them with a warm, wet home, and we feed them the fiber we can't use. In return, they synthesize the vitamin K that prevents us from bleeding to death from a paper cut. We think of digestion as something we do, as a solitary act. But in the end, we're not a single organism. We are a walking ecosystem, relying on billions of tiny non-human organisms just to survive the day.
A very humbling thought.
Thanks for diving deep with us. Go enjoy a glass of water, and take a moment to appreciate the power of osmosis. We'll see you next time.